welcome to the second Gene Club. It gives me a great pleasure to introduce our speaker today. So Dr. Hei-ming Lai is a clinician, scientist, entrepreneur who leads a multidisciplinary team in developing 3D biological tissue mapping methods. He obtained his MBBS degree from Hong Kong U in 2018 before joining CUHK as clinical lecturer in 2019. He is now a clinical assistant professor in the Department of Chemical Pathology of CUHK. His work focuses on developing accessible technologies for stereophenotyping, the study of biological traits in 3 dimensions, notably the invention of non-destructive tissue clearing, deep and uniform multiplex 3D immunostaining, which were published on multiple reputable journals like Nature Methods and Nature Communications. His work on 3D histology has attracted an aggregate funding of more than $6 million over 5 years and led to a Croucher Innovation Award as the youngest and the only clinical track recipient. In 2023, he won 2 gold medals at the 48th International Exhibitions of Inventions, Geneva, one with congratulations of the jury. Dr. Lai's aim is for the technology and database to be accessible by any laboratories globally, to catalyze the exploration of biomarkers and biology in its native 3D perspective, and directly benefit patient outcomes by improving survival and reducing unnecessary treatment. So the topic of his talk is spatially resolved molecular pathology in 3D for precision phenotyping. Dr. Lai?
I changed a little bit of the title, but it remains the same. So we are still deciding for a kind of a disruptive technology that what we should aim it and name it. So is it a phenotyping platform? Is it a stereophenotyping platform, or a solid-based diagnostics? But yeah, we do 3D tissue analysis because I was just not satisfied at reading a book by only 1 page, but I want to read the whole volume. So yeah. So a little bit about myself, because I was relatively young. So when I was in high school, I was an amateur chemist. So basically, I do experiment at home. I bought the chemicals from Art Liu Gai, and then I experiment at home. So luckily, yes. So that's a science nerd, right? What a science nerd would do. And there are some accidents, but it's definitely helped me to build experimental intuition and also to learn by experimentation. So yeah. So luckily, I got into medical school as the lowest achiever that time, and then became a house officer, and then become a clinical lecturer, and so forth, so forth. And in 2023, the technology, I think, is finally ready for us to commercialize or to put into clinical practice as a start. But yeah, talk about the amateur chemist. So we are probably this kind of amateur chemist type. We hack things. So this is what happened one time. And there was a explosion, so it was completely an accident, of course. And luckily, my father still allowed me to continue my experiments after this. And I didn't get myself killed, so that was the near-death experience for me. But yeah, it tells me about safety issues. Anyway, but that time also I was in high school, right? I was learning about this DSE molecular biology. This is the central dogma of molecular biology. So there is a genotype, and then there is a phenotype. And genotype, we have very good platforms to access it. So I think by 2008, everything is already set off for next-gen sequencing, right? And but phenotype, not so much. So I started asking the question, why are we not using a lot of technologies to access the phenotype? So, and then I got into medical school in 2012. So that time, the teacher will always ask us to imagine this is a 3D, 2D plane. So that was kind of a paradox to me because the illustrations was obviously 3D, but the pictures were 2D. So in fact, actually 3D should look like this, right? So it should be able to scroll through different views in different orientations, and you see much deeper layers. So this is one of our main goal. We want to see deeper layers, and we want to see the whole picture. Right. And then that time when I was year 2, there was a paper called the Clarity paper. So it basically turns things transparent. And then with mouse, there is a transgenic mouse that you can label the proteins very clearly. So you have a fluorescence microscope, and now you can look into the brain like never before. So they call it a window into the brain. And that time I was already attaching into some labs in Hong Kong U, saying that I want to replicate this technique. And I never got it to work. And then afterwards, I thought it was my problem. And then I talked to other researchers. They also didn't get it to work. So not working consistently, especially in human tissues. So, but that got me thinking. I did got one time to work in after improving the method, is that if we can image the tissue in 3D, we got a much more complete tissue level. So for example, here is a tumor. If we section it along one axis, we see this kind of plane that there is 2 markers that you can have a drug to act on. But if we slice deeper, both of them are gone. And if we slice a little bit biased, then you still get 1 marker that is actionable. But if you slice it along the different orientation, you get a completely different picture. So this is called tumor heterogeneity. And depending on how you slice it by the technicians, your outcome can be very different. So kind of the patient's outcome or the treatment options is completely dependent on the technician's mood or the machine's performance that day. So this is not very acceptable. So what actually the whole thing is this 3D tumor that we should actually observe. There are clusters of cells scattered around everywhere. And at the cellular level, so these are cells. So usually the red blood cell is kind of a ruler. It's like a 7.8 micrometers in diameter, or you have a cancer cell that is 20 micrometers. And usually when we cut it into thin slices in clinical, you only cut 5 micrometers, not thinner, not thicker. So the resolution will be very rough, because if you only have 5 micrometers, everything will be blurred into a single dot. So it's like blur into this kind of dot. And because that tissue was in a kind of a thin slice in wax, you can't cut a candle into very thin slices. So the resolution is kind of limited. If we can somehow turn it transparent, now you can use optics, and lights can section at infinitely thin dimensions, right? You can just shape the lights instead of physically sectioning. So that would give us a huge resolution advantage, 10 times more, in fact, at least. And then the third one is digitalization. So nowadays, pathologists still look under the microscope and see which slide, this one is the cancer diagnosis or inflammatory diagnosis. But without digitalization, you can't access AI tools, or you can't just share the images, which is more contingent during COVID. And you also can't quantify. You can't use image quantification algorithms to quantify. For example, now we have HER2 scoring for tumor diagnosis. And if you can digitalize it, now you can access quantitative and AI analysis. And then the fourth one is multiplexing. You can simultaneously detect multiple markers, including, for example, whether a cells can have 2 drugs to target them all together, or you can just segment the cells, meaning to classify out the cells much more accurately, which is not possible with current 2D histology. So this is some of an example, but I won't go into details of it because we will have more of it later. Yep. So, and then when I graduated from medical school, and basically I studied enough biomarker profiling technologies, so there was just 2. So if you have a binary biomarkers, for example, whether the tumor has a mutation, like EGFR mutation, then you can basically just mash up the whole cancer, and then you can get all of it, but all mixed up, so there's no context. But it's good for 0 to 1 mutations, right? If you have a mutation, you will detect it with PCR and then next generation sequencing. And then the second one is the section-based. So you sample some of it, but you have the context. You have the more of the tissue view, and also at least you can interpret there is some expression of marker around the tumors. There is PD-L1 around the tumor. There is a lymphocyte, but the sampling errors is huge. So what we think is that we can create some kind of quantitative biomarkers. You have a solid base, so you have all of it, and also all in context, like how many cells per volume, how many cells of those are expressing a tumor marker, and how many of those are actually targetable. What's the response rate is going to look like? So throughout the years, I thought about what should be the ideal pipeline. It's all chemistry. So if you give me a sample, how can I best process it? So what would be the ideal pipeline looking like? So it should be spatially, I mean, the whole sample should be comprehensive. you should be completely scan them. And also you have to be molecularly comprehensive. You survey all the markers that is relevant. The second one is to be quantitative. So, I mean, you sample more data, there's no point in not quantifying them. The third one is to explain them. So if you have a whole book, not just reading a single page, then the whole thing should turn out into a story. For example, like reading immune cells around the tumor, but not entering it. Probably something is blocking their entry and killing the tumor cells. And then ideally, you also scan it like a CT scan. So the patient comes out unharmed, the tissue also comes out unharmed. You can go for it to do the downstream genotyping or electron microscopy. And then, of course, it has to be pragmatic. And in medical school, I learned to be very pragmatic, and also as an amateur chemist, because everything should be low cost and accessible. So in histology, we go back, we have a sectioning, staining, and seeing. Now we can just turn it transparent, and then we can do 3D histology, right? Usually, in practice, we'll swap these two. So we will stain the tissue first, and then we will turn it transparent, and then we can use a specialized microscope to scan it. Turns out tissue clearing is very easy. It was invented like 100 years ago already. So this one is called a old method called BABB. We're still using it today, and it's still the best method out there, despite hundreds of modern advancements. So for example, this one is done in 1914 by the German scientist Spatially. It's still in the museum, so it's quite amazing. And so that's what we will use. And then there is light sheet microscopy. You can do optical sectioning, right? You no longer need to really cut the tissue out with a knife. That is also invented exactly 100 years ago by Zsigmondy. Zsigmondy won a Nobel Prize for this one. So it was, that time even laser was not invented. He's just using sunlight and can do this kind of light sheet microscopy. Modern days, we have a more sophisticated lens, but the basic principles still stay the same. And this is some of our builds, so we build our own microscope, so it's just the same. Except the optical parts may be a little bit more advanced or more high quality, and we use lasers instead of sunlight. Yeah. So we can sculpt the laser into a sheet, and then you can section the tissue virtually. And then, of course, we have all these machine learning tools that we can access now. Previously, it was not possible. I mean, 5 years ago, without machine learning, I think the data is simply intractable to analysis. We are talking about 10 TB per sample, so 10 terabytes. So it was difficult to handle, but now we have compression algorithms and also analysis algorithms that can do it within 30 minutes or so, so which is very amenable now. So now we have this, any kind of formalin fixed tissue, conventionally fixed, we can just stain it, we clear it. We have a microscopy built, we have an image analysis pipeline that we wrote our own codes, and then hopefully we can achieve some kind of meaningful clinical impact. And of course, we return the tissue because we don't have the luxury of cutting one more slice out for another analysis. So the whole thing must remain intact. So that's actually the difficult part, because if the tissue is like a 1 in 1,000 biopsy taken with CT guidance, then the patient is not going to agree on taking another biopsy, which is risky, right? So we have to scan the whole thing and then return for genotyping. So phenotyping first and then genotyping. Right, and then we saw that these three has already been achieved, but the thing that is most difficult is actually the staining. So that got me struggled for nearly 10 years. So I started working on this on 2015 when I was year 3, and I still wasn't able to solve it by that time. So it was the major roadblock that set me from like translating to clinic or starting clinical studies. And the challenge is because it's about how to marinate an egg very deeply and also uniformly. You're not just wanting to get the flavors on the outside, you also want to get it uniformly. So not just a good penetration, but also at least a little bit deeper inside the egg. And also you have to be uniform so that your data is not biased. So we talk to a lot of clinicians. Clinicians are very demanding. Users are also very demanding. The problem is you don't want them to destroy the tissue, so you don't want to punch hole through it for them to penetrate. You don't want us to modify the antibodies, which is proteins, which is very sensitive. They want no specialized machines so that it is cheap, is accessible, is scalable, but they also want it to be fast. So you can't use electric field, you just want simple diffusion but fast. So it's like breaking the physics of the universe. And at the same time, they want uniform across depth, of course. So that time, it was really a struggle. But of course, we solved it. We really dug into the physics. We came up with a model borrowed from geography. So I didn't study geography, but it was a model talking about heat transport. So we can model so for example, these are the antigens in the tissue. So the concentration of the antibodies at any space and time point is equal to the diffusion into this or some convection, but actually minus the reaction that would trap them. So for example, the antibodies are defined to bind to the antigen, so they get trapped on the surface like here. But also they have non-specific bindings and also denaturation and aggregation. They bind to each other. So we know that there is all these kind of factors. So how can we do it? So imagine there is a magic chemical. The magic chemical will inhibit all kinds of protein-protein interactions, but not destroy the protein. Then it's like blindfolding them so that they won't find their partners and got trapped on the surface. So they can just move straight into the room, like not getting handshake with anyone. And then afterwards, we unblind them deep inside the tissue so that they can now bind to the nearest partner. Now you will get a uniform and deep penetration. So that's the goal. The problem is the magic chemical, right? So we study the physics of how solution works. So it's called statistical mechanics. So we use some chemical intuitions, which I'm not going to go into details here, and then search through the databases because we are very low in budget. And we eventually find out this compound called the boron cluster. So it was discovered in the 1960s for nuclear waste treatment, but it has never been applied in biology. But it so happened to have the properties that we want. That is, it can inhibit all kinds of protein-protein interactions, but without causing them to get degraded. So this one is the ideal chemical compound. So we have some experiments. We show that it inhibits all kinds of protein-protein interaction. When you add it, the affinity just go down by various methods of assaying. So, and then also they are not denaturing. So after adding that, the concentration of the aggregates just don't change. There's no more of the aggregates. And also even at the secondary structure level. So this one is a general biomolecular property that we discover. And then we need to do experiments to quantify it, right? So we use, because we are again low in budget, we use the SARS-CoV rapid antigen test that is left over. So that is basically a reaction diffusion model. So if you add more of this compound here into the sample pad, but not the saliva, then more of it will get passed through and then stain less on the control line. So you see this inverse relationship. It's like a simulation of the egg. So you add more of it here, and then it just stain less and less, and then it pass through right here. So it kind of verifies our model is true. So now we can inhibit these kind of protein-protein interactions. Can we unblind them, right? We still have to unblind it somehow. So when I was an amateur chemist, I was reading a book called Supramolecular Chemistry. It was totally advanced and irrelevant. It was not possible to perform that kind of experiments at home. But I somehow read it, and it's kept at the back of my mind for like 15 years. And then it turns out it is now useful. So it can complex selectively this kind of molecule, this cluster. So it's completely selective, not affecting the biology. So both compounds, you can eat it and not die. They are completely non-toxic, and they have this very, very specific reaction. Now you can unblind the proteins so that they can interact with each other. It's like snapping your fingers and tell them to find your partner nearest. So that's how we implement it. So it's very easy. It's just saline and add all those kind of antibodies and add this boron cluster into solution A. Now you have your everything that is diffusing into the tissue very quickly, and then you have your solution B, and which is the complexing agent, and it's just saline, so nothing else. Of course, we did some simulation. So if we add this compound, you just got diffusion so quickly compared to the not control. And we can see the experiments directly. So this one is a whole mouse brain scanned through entirety. So it's like an egg. So for example, here you see all the stains are accumulated on the outside, so and not getting any signals inside, like the poorly marinated egg. But here we are only using 3 days of staining compared to 4 days, but it diffuses even deeper into it, and everything localized perfectly. So it's just a salt and a simple in situ reaction. So this is great. So it even works on a centimeter scale with a deep dive into the physics. And then we kind of have some metric to standardize it. So imagine your tissue is like a sausage, so a hong chang, like a salami stuff. If you stain the hong chang, if you stain the sausage, you will get the stain only on the outside. We are using a lot of food analogy here. But if you cut it, if the staining is not perfectly penetrating, your sausage would not be marinated evenly, right? You have some kind of grayer areas inside. And then you can verify whether it is correct by staining the same marker, but with a different color. And you cross-compare the two at the section level. So here is the unevenly stained channel. Here is the gold standard in green. So we did that on a mouse brain. And then you can see if you have good penetration, you will see the pink and the green completely overlaps. So the pink just go all the way through. But this is poor penetration. You see only a sausage with the outside flavor, but not in the inside for the magenta color. So we actually benchmark all kinds of proposed methods there. And of course, we turns out on top. So we are at least 4 times faster than any available method, and up to 40 times faster than any available method. And it is nearly ideal if we can quantify every single cell of them, and they still turn out to be nearly perfectly representative of the signal. In fact, it is so representative that it is even semi-quantitative. So on the x-axis, we have the standard. On the y-axis, we have the inside our method stain signal. So they kind of get this diagonal perfect correlation, near perfect correlation. And the good thing, it is scalable, right? We don't have any specialized machine. It's just at room temperature. So we just use this plates. We just dump all the organs in, and then we can just stain all the adult kidneys like this one. And as I've said, the denser the antigen, the more the difficult it is sustaining. But here we have a very dense antigen expression. So everything is positive. Supposedly, it is very difficult to get the deep penetration, and you will only get the rim. But here we are seeing all the tubules all at once in this. This one is an adult mouse kidney. So there's nothing to hide here. So previously, people would usually publish only a 3D pretty rendering, but we are not kind of scared to show the scroll through of the whole thing because we stained it well. So it's a nice thing to verify that it worked. And in fact, if you can process large samples, for example, here we have half of the human brainstem, we can map it all the way from MRI to the histological scale to a phosphorylation of alpha-synuclein. So you can go from the micrometer level to the centimeter scale. So everything can be correlated across scales. And of course, if we want to do developmental biology, you can see the nerves, because nerves are pretty, if you cut it, it only becomes a dot for a string. So of course, a video is better. So here is a mouse embryo. Near term is around 2 centimeters in size. And we don't need to remove the skin or the bones. So everything is intact. We didn't cut a hole in there. And the antibody just goes in because of the chemistry. And it is very uniform. And yeah, as we can see, all the medical knowledge just start annoying me now. So the brachioplexus or something. So when my student imaged this, I told him, if this has come out earlier, I might not have failed my medical exam. But apparently it was too late, so I had to endure some retake. Yeah. But anyway, I passed. Yeah, I do qualify. Just a disclaimer. But it was so nice to look at. So I can look at this all day. So for example, here is the cochlea. There's a spiral ganglion. It's like a spiral. So, and this is the superior cervical ganglion. So now I'm well versed in the neuroanatomy, like nobody else, because I see the whole thing. Yeah. Yep. And of course, you got more pretty structures. This one is an adult mouse liver, so a whole one. So anyone has worked with mouse before will know that the liver is the largest internal organ, and we can scan the whole thing, so not even close. Yeah, I think this is the largest mouse specimen ever imaged with simple diffusion and staining. So the mouse liver actually looks a lot like the human liver. Yeah. Again, like a CT, we can scan through it slice by slice now. Right, so yeah, good teaching material for medical students. Now, this one is a human colon. We are now moving into the human arena. So we can see all kinds of structures. For example, the sparse lymphoid patches, which is easily missed. In a tumor, this is also relevant for the outcome of the patient. But now we can quantify it very easily. So we have the mucosa, the submucosa, and the muscle layers. So everything is seen clearly. Right. And then, of course, we have layer structures. We, of course, have the skin. So this one is the skin. We have the epidermis, the dermis, and the subcutaneous lymph node. So even skin as fibrous as it is, and sometimes you have some dark colorations, we can still turn it transparent and then stain it well. So we have a sweat gland here, really a duct of sweat glands that will secrete up to the skin. So yeah, this is the true morphology of the tissues. Yeah. Right. And if you want to look into more delicate structures, just now it's just a gross overview. You can even do delicate structures. So for example, we studied nephrology or the kidney structures. We found that there is a space between the glomerulus and also the parietal Bowman capsule. We found that there is actually this kind of small filaments that is connecting the two. So in 2D section, these are going to be destroyed inevitably. But now we are discovering these new structures that is never described in the literature, because it's non-destructive 3D imaging, right? So these type of structures were kind of like branched, and they seem to be a intercellular context, not a humoral one, but a physical one. So it gives you an example of how we can discover new structures for this one. And if we want to even see better, we can even do epigenetics. So down to the atomic level, the monodi or trimethylation of the epigenetics can still be profiled in 3D, or even the symmetricity of arginine dimethylation. And this is especially good for a human glioma. So human glioma depends on the epigenetics for the prognostic scoring. So this also works. In fact, because our technique is very scalable, we verify 350 of the antibodies. So it's at least, I think it's 5 times more than any other technique out of them, because we just want to be sure that it can work. And it can also be multimodal, so we can simultaneously stain proteins, mRNAs, epigenetics, glycosylations, and nuclear morphology all at once in the same sample. So got correlated multimodality analysis. So how about preservation? So we have this kind of control sample. We put it in the fridge for storage as a control. And then we have this tissue that is virtualized and 3D scanned. And then we subject to some kind of rigorous test, and then see whether pathologists can tell them to guess which one is 3D processed, which one is not. And turns out they can't do. They just guess a 50-50. So it's indistinguishable by pathologists using H&E or IHC. And we even do a whole genome sequencing. The whole genome sequencing is still intact and it's not changed. The bulk transcriptomics, even RNA, remains sequenceable, and there's no differentially expressed genes, so it's also intact. And at the proteomics level, it's also not changed for 7,000 proteins. Usually, we're only getting 1,000, but the quality is so good that we get 7,000. So it's completely unchanged. So this gives us new ways to find out biomarkers in the tissue that is previously not possible. For example, here, again, we have a pretty picture of human colon cancer that we see the nerves, the cancer nerves, and also the vessels. But more importantly, this sample actually was sent to Bruker nanostring to re-embed in paraffin and cut a single slice out to do spatially resolved transcriptomics and also proteomics. And they are still able to do 1,000 microRNAs, mRNAs, and also 64 proteins. And we can map it back directly to the 3D optical section. So everything is correlated. Everything is not destroyed. So it gives kind of a 3D reference to the multiomics screening of biomarkers, which is especially important, I think, at this age, because we now have OMICS technology, and you can use 3D to do it as a reference coordinate space. So I think we kind of really solved these issues, and we really did achieve deep immunostaining, and also we were able to return the tissue that you can't tell whether we do something about it. So, and the whole thing is standardized. So all the tissues that you saw in terms of images, they were using the same protocol. So no need for re-optimization. They were just using the same one. so which is essential for clinical use. And of course, people still ask questions whether they can, can you use archival specimen? Like 30 years ago, I have some very precious cohorts. Can you use it again? So that was, again, a very difficult challenge. Because FFPE, if we know, it was usually 2D, it was not doable with IHC because you have to do this kind of antigen retrieval. So pathologists before 1991 is not, you can't just correlate the two. Until this little known guy came along, he published a very small paper that would revolutionize the whole pathology industry. So what he created was antigen retrieval, that you can do immunostaining on paraffin and beta slides. So you get both morphology and immunostaining. So we want to achieve that in 3D as well. So can we achieve that? So we solved this in, I think it's just a month ago. So for example, here we have a fresh control. This is a human colon. If we use the conventional 2D method, you just can't get the retrieval, so you don't get this kind of staining. So 2D methods won't work. But we have developed this kind of new technique called rescue. Now you can stain back all the markers that you want to stain. And in fact, it is so good that it is indistinguishable again, whether it is embedded or not. So you can have a guess, like which of them are rescued or which of them are fresh. For example, here we have other antibodies. And I'm going to skip this, but usually we'll give a test on the audience to tell whether people can guess it or not. And in fact, you can go back really 1, 10 to 30 years. For control, if you don't rescue it, you can stain nothing. But now we can stain back the markers for this red marker here. So it proves that we can go back in time and dug out the archives. And again, it is our style to not just verify at the morphological level, we verify it at the OMICS level. So we do 3 samples, control 1, 2, 3, and then compare with test 1, 2, 3 at the proteomics level, and they correlate very nicely with each other. So the process was non-destructive. And so there's only around, I think it's 2% of the proteins that is downregulated beyond half. So PCA do show some distinguishability, but I think it's quite okay already. But we'll try to improve that a little bit better. And that would kind of open up to kind of a combined assessment of actionable phenotypes. For example, here we have a 1994 non-small cell lung cancer, which is as old as me. And then, of course, we can see other cases. For example, here we have a mutually exclusive expression of the 2 markers, which means that you can target the same disease with 2 drugs for this patient, because it's multiplex, right? We are showing only a portion of it here. Otherwise, it would be too complicated to show. And then now we have a complementary one, so which again suggests of the dual use of the therapeutics. So what we are doing is trying to run a retrospective study to define the scale and the imaging parameters that would lead us to a companion diagnostics. So of course, for immunology profiling, we want to see all the immune cells, right? So here we have a human colon with all the immune cells mapped out, different types of it, just to show that we can do this. And we have human appendix, FFP retrieval, but this is just to show that for the researchers who are not convinced, we show that we can do this because the image just can't lie. So something like that. So a kind of a summary. So we now have any tissues, whether it is fresh or archived, we can do kind of a staining and then somehow scan through it, and we can get 100% of the information out of it. You get a 3D perspective, you get structural molecular correlates, and also spatially resolved single cell statistics. So we can show them a little bit later. And the good thing is that there is no difference by pathologist examination, so you can be backward compatible with genome sequencing, transcriptome, or even proteome. And so this kind of gives us a opportunity to virtualize a tissue. So we can just literally do a CT scan on it and then store it in the cloud, leave it for database for future analysis and sharing with 1 million times more information. So not just digital pathology. And then if we want to discover more biomarkers, apart from doing in 3D, we can go back and section it and then do some, again, omics studies, especially with spatial transcriptomics. So you can now understand them in more context, and which is now a kind of a demand from the spatial transcriptomics community because they just can't reference where they are cutting the slides. So I can illustrate some of the quantification that we've done. We only see some pretty pictures, right? But they are useless. So this is what my supervisor told me back when I was in my university lab, so in my undergraduate. So that was very harsh, only pretty pictures. So we took this liver that you just saw, and then what we can do is that we can stain the membranes and also the nucleus, but we can just focus on the membranes. So we key out the nuclei. So this will create the boundaries for the cells, and you can annotate each of the individual single cells here. So that each of the mosaic is these mosaics is like each of the color is individual cell ID, so you can key them out. I think this is not showing very well because it's sharing through the Zoom, but the scrolling is not smooth. But anyway, we can now quantify it. So we can quantify, here only you have 15,000 cells, not that much. And then because liver cells are known to have more than one nuclei, and textbooks says that it can only have up to 2 nuclei. So we question that, we just quantify all of it and see. And then we found some very rare cell types. For example, here is obviously have 4 nuclei. So this one is already above the record, right? In 3D, you can see everything with clarity, but in 2D, maybe you would doubt, or maybe it's just a sectioning artifact, you're just missing something. Is your angle correct? So they never was confident in making this kind of statement. And if we can classify them, we can classify like 4 layers, right? Here is the 8 nuclei, 4 nuclei, 2 nuclei, and 1 nuclei. There's no 6 or 7 nuclei. And if we simulate by cutting the same tissue in computer, in cutting 9 sections, we get a similar number of cells, but you don't get this kind of layer structures. So 2D sampling is just not representative of a 3D one. So if we want to quantify the expression of a biomarker, for example, a cancer cell, you want to quantify whether it expresses a HER2 that is targetable by a drug, we can quantify it by the number of signals, the amount of signal per surface area. So it's a 3D object, it has a surface area. We get this kind of clear bimodal distribution, so we can just define and cut off, oh, here is the expression high one, and here is the expression low one. If we again do that with 2D, we see that there is no bimodal distribution because we are quantifying it by signal per perimeter. So it's different, right? It's a 2D, you only get a perimeter of the cell membrane. Even if you sum them all and do statistical averaging, they are simply not the same. So you don't get this kind of bimodal distribution that you can readily define and cut off because it's simply not the whole thing. And if you want to extend along the dimension, so here we have a 1,000 micrometer thick tissue. We can quantify 28 markers here. And of course, these are just the quantified staining images shown. But we can really get all the cells and then segment them in high dimension. So it's a 3D spatial resolved bioinformatics analysis, which other people can only do 2D as of now, but we can now move on to 3D and with morphology. So it's a demonstration of what we can do. And of course, we quantify all of it. We are going to skip this because this is for neuroscience community. And then for lung cancer, so we have this lung cancer, we can quantify the HER2 level. So these are like the cancer cells keyed out, and then they are like pebble stones here. And then if you have higher level, we color them by different colors. But you can also quantify how far they are from the vessels. And this is actually the previous lung cancer sample that I showed you. There's 1 million cells here, so we can just quantify across the sample. And if we do virtual sectioning and then try to compare an X scoring and in 2D section, we can see that the ground truth for 2D will never meet the ground truth for 3D. So 2D mean is around here. So even if you cut 1,000 sections for the same thing, it will not meet the 3D ground truth. And it is statistically significant. And if you cut thicker sections, like maybe you are saying, 5 micron section is too thin for a single cell, you can cut a 50 micron, so that would encompass the whole cell. And the result is slightly better, but you are still missing the ground truth. So no matter how you cut it or simulate it, it's simply impossible to represent 3D structures in 2D. So we actually did some quantification. This is in collaboration with AstraZeneca. So we try to quantify these 3D cell expressed biomarkers, and then we can calculate the score of the tumor cells expressing these kind of biomarkers. We define the score based on some metrics, and then we found that, of course, again, 3D and 2D have a very high disparity. In some cases, this is the 2D mean, it completely misses the 3D one because the tumor is just so heterogeneous. So I'm kind of repeating myself here already. What we are actually doing, we want to make the point is that we are trying to take out all the cases and then redefine the diagnosis to see whether they will actually have been fit for some kind of therapeutics. So we are collecting 180 cases of non-small cell lung cancer. And then we try to define what would be the diagnostic look like how much should we image and what is the pattern that we have to look for. So I think I'm going to sum up now. So for diagnostics, the goal has always been to picture what is accurate, what is actually wrong within you. So if it is not representative of your biology, then it is quite meaningless. So previously we have 2D, X-ray, and now we're moving to CT, and you can get more accurate of a picture. And again, we have the genotyping platform. We get much more information compared to just doing a PCR, which is just a single gene fragment. So I think the point here is that the information density. So if you have X-ray to CT, you get much more information. And going from PCR to sequencing, again, you get much more information than just a single point sampling. And I think 2D to 3D histology here, or whatever we are going to call it, like from section to solid, then you are going to get much more information. So what do we see ourselves? That's a very good question. We actually don't know because we will leave this kind of technology platform for scientists and clinicians to explore what are the potential avenues for exploration. But I think it's kind of like a comprehensive phenotyping platform. Now it is hypothesis-free. Previously, you will have to do qPCR to target a single gene fragment. Now you can just sequence everything and then leave it to the computer to find out the patterns. It's similar that we can now quantify all the phenotypes cell by cell, and then just leave out to the scientists and also the researchers and clinicians to find out the phenotypes and correlate. So because Dr. Lai wanted me to talk about my personal journey as well, so this all started when I was in year 3 that I decided really to pursue this path. I think the goal has always is to make something that people need. So in commercial world, as an entrepreneur, maybe we are making something people want, but in medicine, we are making something that people need because if we have this, people can survive better or live better lives. So I think 2D histology has been a bottleneck in the technology in terms of diagnostic, and there are evidence to show that, which we can discuss. And the approach over the years, I mean, there is a lot of noise these days. Like there has been awards, there has been award honors to hope to drive the biotechnology or innovation industry forward. But I think those are all secondary. So really what ultimately drives me to do microscope late at night is really what I want to see. I only want to see a beautiful picture of neurons or tissues. So that is irreplaceable by itself. So no matter how much the drive is, it's not going to cut it. And then our team's motto is very simple. There's nothing is impossible, it's just learn it. So for example, building a microscope, nobody knows how to build a microscope. It was not taught in medical school, right? The optics, the physics, and the coding. We just learn it from YouTube. So, and then we just buy the thing and then we experiment. We can break something, it's gonna be costly, but that's what money is for, right? It's to experiment. So that's why we just learn everything. We do the chemistry, nobody taught us, we just learn it ourselves. And speaking about learning is that I think the biggest lesson I've learned through a quite short period of entrepreneurship is to talk to our customers, because we will never understand what they need or what the product is need. So as a doctor, we will kind of usually be an authoritarian status. So we know what is best for our patient. Of course, we can talk to them a little bit, but when it comes to developing a product, we need to know what is the real situation there. So to this day, I'm still attaching oncology clinics as a medical student, just to see how the oncology environment would run. I will attach to oncology grand rounds just to see how they make decisions. So to make our pipeline not to get off the ground. So because scientists usually will get off the ground and become utopians rather than grounded to earth on developing more pragmatic and realistic technologies. And then there is these lessons of patents. So I filed my first patent in 2016. That was still in med school, but that was kind of due to a stupid reason, because I thought that someone is going to copy us. And patent is not a thing to avoid copying. Patent is originally to help you to exclude others from profiting based on the same idea. So it was developed in Venice some like 200 years ago. But it has evolved because it is a legal tool. It has evolved to become a commodity for negotiation, a tool for discussion, and an asset to trade. So it's kind of like, unlike publishing scientific papers. Scientific publishing paper, you can only put it on your CV and hope for a job. But patent is a commoditized knowledge, so that you can actually profit from it, but at the same time get harm from it, because it's like your dual. It's like a friend and also a foe, because it can be a liability. You can push a large sum of money in it, and 99% of the patent won't turn out. I had the opportunity to learn from the best. So these three guys were very kind, and they really taught me step by step like how to do it. So there is a lot of know-how that is only earned through experience. That can only be learned through experience, because it's dynamic. So it depends on how people use it. So someday there will be some innovative use of patent to attack you, and then you can use another patent to defend it. So it's like a dynamic evolution of weapons, depending on how creative people are. And then there is this thing called determination. So it's the drive that we want to make 3D imaging work, but nothing in the literature works for us, so we have to develop our own. So in our team, we have this kind of saying that we have no unconditional love for our technology. So that means we only love our technology if it works. If we found out a literature works better than us, we use the others. So we are not owning the technology. So we don't love our products, we love our customer. So as long as they can use it easily and use it best, we will ditch our product completely. We will go for the other one. But the thing is, we are kind of putting a lot of things behind, because we have to be laser focused. So I kind of transitioned from a doctor, which is a very stable position, and I ditched the specialist training. I almost ditched my professor responsibility. Thank God I wasn't fired. And also chasing the short-term awards and those kind of work-life balances. But instead, we go for debt, we go for risk. And there is some kind of loneliness in here, because it's not like Silicon Valley here. So there is not much peer that you can talk to about the risk and also the struggles that you're going through. And of course, there are many uncertainties. To this day, we don't know whether it will play out. And people talk about love your product, but we actually really love our customers. So we really go to conferences and then find what will work best for them, and we try to develop the technology for them. But yeah, that's the story for another day. So final thing is about execution. So there is a lot of great ideas out there, but the idea is not even patentable. So it's only through execution that you really bring the things to life. And there is a huge ton of struggles through it. So I think everyone here in the room who has worked through a project would know. So yeah, and I think Steve Jobs really put it very nicely. So I really love the quote here. So, and then I leave a final question for the audience, is that so far my career has been very unshiny. So I was just focused on this very little thing that I want to do well. And luckily, I have received some support from some very understanding bosses and also friends who can allow me to dream continuously. But I think there is a need for building an ecosystem. So I wanted to let the world know that Hong Kong can also build this kind of disruptive technology, that when we go to conferences, people will be amazed at how come there is a guy who I've never heard of, not from Harvard, not from Stanford, who have solved something that even those people can't. So yeah, so I hope there can be more initiatives and also the correct drives for Hong Kong to be a hub for innovation and technology. Yeah, so we can aim at something else rather than on secondary drivers. So after all, this project was originally driven by really love 用開發電嘅係啦。So it's not based on money or profit or honors, but really what we love. Yeah. So thank you very much for the attention, and so I will take any questions. And thank you for inviting.
Thank you very much. Thanks a lot, Hei-ming. So definitely beautiful work. I think it's beautiful visually and it's beautiful scientifically. And really, you put yourself as the real life answer to the last question that you place. Can Hong Kong do disruptive technology? And your work certainly testify that we can. And this is really an innovation that drives medical revolution, because we're seeing the change from X-ray to CT scan to RNA to MRI, and then we're seeing PCR to whole genome sequencing. You're really leading the way from 2D histology and 3D into 3D histology. And I think the medical textbook is going to change because of your work. And the students is going to study more before the examinations for your work.
I was actually very struck by your comment about your life journey of facing the 10-year roadblock. I want to ask you, what was your emotion like for that 10 years? And secondly, how did you stay focused on what you wanted to achieve? Thank you.
Yeah, so the story goes like when I was in year 3, like 10 years ago, exactly 10 years ago, 2015. So I was turning things transparent, and I start to realize that, you can't just stain them deeply. And it was, to say the least, it was frustrating, right? But I already have been pitching my dreams to others, like my fellow student who would not, my fellow classmate who would not understand why the fuss, just study and get a distinction, right? And then there is this huge expectation from others to hope that you can deliver, like saying that you should develop the deep staining. And you never know whether it will play. I mean, because it's chemistry, it's science, not engineering. You can't have a formula to calculate out the probability that this will work out. It may not exist a solution in the universe at all. Until I found out that kind of boron cluster compound, which is kind of out of the blue. And so the anxiety, I think, is really, really huge. So, and I think what makes me persevere is that I really just want to see the deep image. It's just that. So I was not, whenever I see a failed image, like staining as a very thin slice, I was just not satisfied. I just really want to see deeper layers. I want to reconstruct them see it with my own eyes. There was no clear path towards commercialization. I didn't know about commercialization that time, patents or so, publication or so. I just want to see a good image. I just know that literature methods just don't work. And I would put it as that maybe it's my problem, but I was taking other peers that it also doesn't work in their hands. So I must invent a new method. And it was very, if I'm going to pitch this problem to my fellow peers that time, nobody would agree with me because they say that they have published it, it must be good in big journals, but it doesn't work. So I still can't get to see the things myself. So it was this kind of frustration that continuously to drive me forward. I just want to see the thing. Yeah, nothing else. Yeah.
So I just want to remind audience in Zoom, if you have questions, you can type in the Q&A chat box. I would like to open questions to the floor. Is there anyone asking questions? Let me ask the second questions. I was in MBBS program and it is terribly busy. In year 3, how can you still start such a wonderful project that nobody else has been doing? Is obviously something not given to you as an assignment from your supervisors. This must be brand new. And how can you balance your very busy schedule there?
So that time I would wake up at 7 and I would go to the core facility to set up the microscope to image 1 hour before I go to school, and then walk back up to primary hospital to junior clerkship or something. Of course, I did get scolded a lot of times for being late. And then usually at lunchtime, if I was able to book a microscope from morning to afternoon, then I would, during lunchtime, run back down to the Sassoon Road, retrieve my sample, and then go back to lessons. So I would skip lunch for that. And sometimes, because that time the microscope in core facility was very slow, so taking a big image like you saw would take like 15 hours. So what I would do is that I would, at 8 p.m. after my lesson, I would again run down, set up an overnight imaging session. So this was unlike anything they have seen in the core facility. They haven't handled such a large amount of data. That time, if you copy such a data set like 100 gigs, it would take a day. So, and then the next morning at 6 a.m., I would go back again and retrieve the sample because we have to be earlier because saving 100 gigs of data will take 1 more hour. If you can't save it, it's gone. So squeezing time, just that.
Yeah, next time when I have a student that comes late, I would certainly ask
Yes.
for the reason first before I actually make further comments. Why are you late? Okay,
Yes.
so now I'm a more understanding teacher. So the other thing that I'm very interested in is now you have mastered the technology, and I think in your web page you have this dream of setting up a database. I think you're building up certain kind of map so that when different specialists navigate for their own favorite pathology, they would have a reference. How do you envisage that you're going to build that massive 3D histology map? Are you already working on it?
Yeah. So yeah, we're working on this because we have the highest throughput of any other technologies out there. Of course, we want to take advantage of this one. And in the AI era, data is king. So whoever holds the most amount of data, whoever will be the winner, right? It's like having a pathologist or your AI pathologist having access to the best AI database textbook, so you can train the best foundation models for them. So the other thing is we are not chasing the hype. We don't position ourselves as an AI first company. We are AI second company. So we make the data first, and then AI company will try to find us for the data sets to train their model. So because we're not good at AI, we just don't want to chase it. Otherwise, we will lose the competition, right? Yeah.
Yeah, I think we are really thinking the same here in the Hong Kong Genome Institute, because we are also accumulating a lot of genomic data, representatives of Hong Kong, and then AI is really second. We have to make good use of the tool to make good use of the data that we have actually accumulated throughout the year. I would just like to check if the floor have any further questions.
I just want to ask, your image, 3D image, I assume it takes up a lot of data, right? As you said, like even 100 gig for like 10 years ago. So I just want to ask how big is one file of data, one file of image? And is the current technology, like capacity of infrastructure, a bottleneck for your work? Thank you.
Yeah, so how big is one data? So if you have a 1 cm cube human tissue virtualized for 4 markers, that's 1 terabyte now at the resolution that people want. So then brings us to the second question, are the infrastructures ready? I think it's ready. So we were able to send these files to our collaborator in quite short amount of time, like in 30 minutes. So it's okay. The computational infrastructure was just advancing at an amazing speed. So when I was graduating, I thought this was impossible. But then there comes AI and also all those data centers. And then suddenly it becomes possible. Like at that time, I don't know how to code. So, but I was still able to upload my files to my collaborator. So yeah, we can just wait for the others. The best thing about humanity is that they will innovate on their own, but somehow their solutions will be your savior sometimes. Yeah.
Thank you. Perhaps one question from the Zoom is Professor Ip Seping from Poly Youth. He has 2 questions, but before that, great talk, very illuminating talk. So the first question is, what will be the greatest number of proteins that you can study in one specimen? And the second question is it possible to study protein DNA and protein RNA interaction in your model?
So people just keep asking for more
Customers.
Customers, right?
But there are no customers.
Which is good. Yeah, because people are asking questions. That means we have continuous work to be done. And the max amount of proteins we can do is 28. So currently, we have a newer technology coming up that I haven't included in this talk. It's like 50. So, but we still have to validate it. And then about protein DNA and protein RNA interactions. So we haven't worked on that, so that's a good point, and I can try. We are working on protein-protein interaction, so that's called a proximity interaction. So if you see 2 proteins co-localizing, that doesn't mean that they are interacting, because it's just like using a satellite to view this room and see who is shaking hands, which don't have the resolution. But we are building this technology that is called proximity that detection, that we can actually see molecules touching hands and shaking hands. So that will really give you whether a cancer is really primed by the receptor, or there is a binding of the ligand to the receptor. So yeah, so that we are working on that.
And, your answer is so stimulating that the same customer, that is Professor Eve, is asking you the third question. So could you touch on issues like data analysis in terms of the algorithms and new tools that you're using?
Yep. So machine learning is certainly a large part of it. So because if you want to circle out the cells, normally with traditional algorithm, you have to define the cell mathematically. Like if it is a sphere, then you define it by having the same radius over it. But biology is too complex for that. So machine learning is helping us to fit this parameter to segment the cells. So we train our custom models to do that, and we code our custom scripts. There is this good model called UNet, which is very useful for morphological segmentation, and is keeping to pump out good training data sets and segmentation over and over again. Yeah.
Thank you so much. I think although that we have more questions, because of at the interest of time, we will have to close the session here. And the privilege of those who are actually physically participating in Gene Club is we have more chance to interact with you after the talk. Thank you very much, Hei-ming. So before I actually close the session, let me actually once again thank our great speaker, Dr. Hei-ming Lai. And it's also time for me to do the advertisement. We have the third Gene Club that is upcoming. So our next speaker is Dr. Becky Ma. So Dr. Becky Ma is a nephrologist. She received her MBBS degree from the University of Hong Kong, and she was trained as a nephrologist. And she is also have a lot of experience in kidney disease and the related genetics. She has been awarded the Health and Medical Research Fund Fellowship Grant, and also the Hong Kong College of Physicians and Hong Kong Genome Institute Training Grant for Excellence in Genomic Medicine. So the topic of her talk is going to be Hong Kong Genome Project, so it's about us, case sharing, precision medicine in nephrology, and it will be on August the 7th. Please stay tuned for this next Gene Club. With that, I would like to close today's Journal Club. Thank you very much for your kind attention.